Bismillahirrahmanirrahim. The topic of today's lecture is Introduction to Immunology. Learning objectives. By the end of this lecture, you will be able to define and classify immunity, define antigen, define pathogen, enlist the tissues that contribute to immunity and explain their functions, the functions of the immune system, and to describe the structures and function of the lymphatic system. Now coming to the definition of the immunity. The word immunity is derived from immunus, which means to protect or exempt. Immunity is a broad definition. This is a protective or defense mechanism of our body, which protects us from infections. Immunity is the ability of the body to protect against all types of foreign bodies like bacteria, virus, toxic substances which enter the body. As it protects us from diseases, it is also called disease resistance. Lack of immunity is known as susceptibility. Immunity is done by immune system, which is a complex network of lymphoid organs such as bone marrow, thymus, spleen, etc. Now, immunity is divided into two main classes. One is innate immunity, which is non-specific immunity, and the other is acquired immunity, which is specific immunity. Acquired immunity is again divided into two main classes. These are the humoral immunity and the other is cellular immunity. Humoral immunity is antibody mediated, while cellular immunity is T cell mediated. Now the innate immunity. Innate immunity is the inborn capacity of the body to resist pathogens. By chance, if the organism enters the body, innate immunity eliminates them before the development of any disease. It is otherwise called the natural or non-specific immunity. Innate immunity includes the following: phagocytosis of bacteria and other invaders by white blood cells and cells of the tissue macrophage system. Destruction of swallowed organisms by the acid secretions of the stomach and the digestive enzymes. Resistance of the skin and the mucous membrane to invasion by organisms. Presence in the blood of certain chemical compounds that attach to the foreign organisms or toxins and destroy them. Some of these compounds are lysozyme, which is a mucolytic polysaccharide that attack bacteria and causes them to dissolute. Basic polypeptides, which inactivate certain types of gram-positive bacteria. The complement complex, a system of about 20 proteins that can be activated in various ways to destroy bacteria. And natural killer cells, which are the lymphocytes that can recognize and destroy foreign cells, tumor cells, and even some more infected cells. Acquired immunity. Acquired immunity or specific immunity. Acquired immunity is the resistance developed in the body against any specific foreign body like bacteria, viruses, toxins, vaccines, or transplanted tissues. So this type of immunity is also known as specific immunity. It is the most powerful immune mechanism that protects the body from the invading organisms or toxic substances. Lymphocytes are responsible for the acquired immunity. There are two types of acquired immunity: cellular immunity and humoral immunity. T lymphocytes are responsible for the development of cellular immunity, and B lymphocytes are responsible for humoral immunity. Now this slide shows the comparison between innate and adaptive immunity. Innate immunity is active prior to exposure to any microbe or antigen, while adaptive immunity is activated by exposure to microbes or antigens. Lag phase is absent in innate immunity and it gives immediate response, while lag phase is present in adaptive immunity and it takes few days to respond. Innate immunity is limited in specificity and it targets all the pathogens, while adaptive immunity is high in specificity and it targets specific pathogens. Memory cells are absent in innate immunity and it gives the same response in subsequent exposures, while memory cells are present in adaptive immunity and it gives amplified response in subsequent exposures. Now what is antigen? Antigens are the substances which induce specific immune reactions in the body. Antigens are mostly conjugated proteins like glycoproteins, nucleoproteins, and lipoproteins. Types of antigens. Autoantigens are self-antigens, and foreign antigens are non-self antigens. Autoantigens or self-antigens are present on the body's own cells such as A antigen and B antigen in RBCs, while foreign antigens or non-self antigens are those antigens which enter the body from outside. For example, receptors, toxins, material from transplanted organs, incompatible blood cells, allergens, etc. Types of non-self antigens. Non-self antigens are classified into two types, depending upon the response developed against them in the body. Antigens which induce the development of immunity or production of antibodies, and antigens which react with specific antibodies and produce allergic reactions. After discussing the antigens, we should know about the pathogens. What are pathogens? A pathogen is an organism that causes diseases. All pathogen needs to survive in a host. Once the pathogen sets itself up in the host body, it manages to avoid the body's immune responses and uses the body's resources to replicate. Pathogens can be transmitted a few ways depending on the type. They can be spread through skin contact, bodily fluids, airborne particles, contact with feces, and touching a surface touched by an infected person. Pathogens are of different types. Like viruses, once you are infected, viruses invade host cells within your body. They then use the components of the host cell to replicate and destroy the infected cells. Bacteria. Bacteria are microorganisms made of a single cell. Not all bacteria cause infections. Those that can are called pathogenic bacteria. Fungi. Fungi can be found just about everywhere in the environment, including indoors, outdoors, and on human skin. They cause infection when they overgrow. Parasites. Parasites are organisms that behave like tiny animals, living in or on a host and feeding from or at the expense of the host. After discussing the antigens and pathogen, we should now know about the term antibodies. What are antibodies? An antibody is defined as a protein that is produced by B lymphocytes in response to the presence of an antigen. Antibody is gamma globulin in nature and it is also called immunoglobulin. Immunoglobulins form 20% of the total plasma proteins. Antibodies enter almost all the tissues of the body. There are five types of antibodies: immunoglobulin A, immunoglobulin D, immunoglobulin E, immunoglobulin G, and immunoglobulin M. Among these antibodies, immunoglobulin G forms 75% of the antibodies in the body. Organization of the immune system. As immunity has two main basic types: innate immunity, which is non-specific natural immunity. It includes anatomical barriers, physiological barriers, cellular barriers, and inflammatory barriers. While adaptive immunity, which is specific and is acquired when a specific pathogen enters into your body, it includes lymphoid organs like bone marrow, thymus, spleen, and it also includes cells like white blood cells and molecules like different types of cytokines. Now the anatomical barriers, which include skin, mucous membrane, which is the component of innate immunity. Epidermis acts as a mechanical barrier and retards entry of microorganisms. Acidic environment retards growth of microorganisms. Normal microbial flora compete with the pathogenic microorganism in the mucous membrane for the attachment and nutrients. Mucus entraps foreign microorganisms. Cilia of the surface epithelium propel microbes out of the body. Physiological barrier, which is another component of innate immunity. It includes fever response, inhibit growth of the microorganisms. Acidic environment in stomach kills microorganism, in vagina retards the growth of microorganisms. Chemical mediators like lysozyme of tear cleaves bacterial cell wall. Interferon induces antiviral state in uninfected cells. Complement system lyses microbes or facilitate their phagocytosis. Antibacterial peptides kill pathogenic bacteria. Cellular barrier. Cellular barrier includes phagocytic cells, which are neutrophils, monocytes, macrophages, dendritic cells, eosinophils. They detect pathogen-associated molecular pattern on the surface of the microbes by their pattern recognition receptors. They internalize the pathogens and kill them. Then non-phagocytic killer cells. These include the natural killer cells, which induce apoptosis of virus-infected and tumor cells. Now, inflammatory barrier. Inflammatory response upon tissue damage or microbial infection induces leakage of vascular fluid containing antimicrobial peptides to the site of damage or site of entry of microbes. Inflammatory response also causes influx of phagocytic cells, which are like neutrophils, monocytes, and macrophages to the affected site. Antibacterial peptides and phagocytes then destroy the microbial agents. Now, the role of white blood cells in immunity. White blood cells play an important role in defense mechanism. These cells protect the body from invading organisms or foreign bodies either by destroying or inactivating them. However, in defense mechanism, each type of WBCs acts in a different way. This slide shows different types of leukocytes or WBCs. Mainly WBCs are divided into two main classes, agranular and granular. Agranular include lymphocytes and monocytes, while granular includes basophils, neutrophils, and eosinophils. Lymphocytes are again divided into T lymphocyte cell, B lymphocytes, and natural killer cells. Every cell has its own way of killing pathogen. Like neutrophil and monocytes are phagocytic cells, and macrophages, which are derived from the monocytes, are the major phagocytic cells. Natural killer cells direct kill the pathogen. T and B lymphocytes are the cells of acquired immunity. This figure shows different types of WBCs like neutrophils, natural killer cells, macrophages, and eosinophils attacking the target cells. They attack the target cells by their own way. Neutrophils and macrophages are the phagocytic cells. Natural killer cells attack the target cells directly and kill the infected cells. Functions of the immune system. Recognition of self and non-self antigens, maintaining homeostasis by removing old and damaged cells, complex and broad range of defensive mechanisms used to combat infections and diseases. Now, lymphatic system. Lymphatic system is the subset of immune system. Lymphatic system include lymphoid organs, and lymphoid organs are divided into primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs are bone marrow and thymus, which is the maturation site, where B cells and T cells become mature. Secondary lymphoid organs are spleen, lymph nodes, mucosal associated lymph tissues, gut associated lymph tissue, trap antigen, antigen presenting cells, lymphocytes, etc. Now this figure shows the different structures included in the lymphoid system. Bone marrow, which is the site of white blood cell production, lymphatic vessels carry lymph from the tissues to the lymph nodes and lymph organs before returning to the blood. Lymph nodes, tonsils, malt, appendix, collections of lymphocytes for immune response and macrophages to remove debris. Spleen replaces worn out blood cells, collections of lymphocyte for immune response and macrophages to remove debris. Thymus is the site of T lymphocyte maturation. Now functions of lymphatic system. Removal of interstitial fluid from the tissues, absorbs and transport fatty acids, fats and chyle from the digestive system, transport WBCs to and from lymph nodes into the bones. Most of the cancer cells travel through lymph nodes, forms part of the immune system and helps against the foreign invaders. This brings us to the end of this lecture. Hope you understood the lecture. Thank you.